Bismillahirrahmanirrahim. Alhamdulillahirrahmanirrahim. Wassalatu wassalamu ala asyrafil anbiya'i wal mursalin wa ala alihi wa sahbihi ajma'in. Rabbi shrah li sadri wa yassir li amri wahlul 'uqdatam min lisani yafqahu qawli. Assalamualaikum warahmatullahi wabarakatuh and very good morning to everyone. Okay, so I think we finished on chapter 3. Now let's go on to chapter 4, which is antigens and haptens. Antigen and haptens. Okay, so basically in this syllabus, in this chapter, you have to know the properties of antigens, factors influencing immunogenicity, hapten carrier conjugate, and major histocompatibility complex. So in other words, you have to know a lot of terms, a lot of definitions that you have to know exactly what the meanings. Right, so this is the introductions on chapter 4. So both B cell and T cell. Let me change this one first. Okay, so as everyone know, B cell and T cell must be able to specifically recognize unwanted cells. So otherwise, all cells in our body system will be recognized as a foreign substance or foreign antigens or foreign cell. So we don't want it to be happen. So that's why B cell and T cell have to play an important role, so that both of them have to recognize all cells that origins or appear in our body systems. So other material than recognized cell will be destroyed or neutralized as it being distinct from the body's own normal cell. So other than cell from our body, everything will be recognized as a foreign substance. So it will be subjected to be destroyed or neutralized. So then an antigen is a foreign unique molecule that triggers a specific immune response. And so basically there is three terms that describes a condition where immune response is able to be activated. Okay, so the first one is immunogens, second one is antigens, and the third one is haptens. So all of these three basically can, not all, but it can activate the immune system response. Okay, so let's go to the definitions of these three terms. The first one is immunogens. So immunogen is basically a substance that can induce a specific immune response. So it doesn't matter cell-mediated or humoral-mediated immune response, as long as that particular substance can induce the immune response, we can call it immunogens. Okay, and the second one is antigens. So antigens is basically a molecules that interacts with the immunoglobulin receptor of B cell or with the T cell receptor when complexed with MHC. So I'm going to explain MHC later on. Basically, antigen is a molecule that can make a link, so it can bind to immunoglobulin receptor. So it doesn't matter from B cell or T cell, as long as it can bind to the immunoglobulin receptor, we can call it an antigen. Okay, and last is hapten. Hapten is a small molecule that is incapable by itself of causing an immune response. However, it can when combined with a carrier protein. So hapten is like a dependent molecule, small molecule. So you can see here figures here. I put it here. So when it's appear only hapten, so only one single hapten, it cannot trigger an immune response, but it can bind to the immunoglobulin receptor. So it means that the receptor from T cell or B cell can recognize hapten. But then, when it recognizes only haptens, no immune response can be triggered. Okay. Otherwise, if it binds to the carrier protein, a larger molecule, basically proteins, then it can induce immune response. Okay. So I can say that all antigen is immunogen. What is the word? Okay. All immunogens is antigen, but not all antigens is immunogens. So if you can understand it, because hapten is basically recognized as antigen, part of antigens, but it cannot trigger the immune response to be activated. Okay, so we cannot call haptens, a single hapten is immunogen, even though it's a part of antigens, because it's foreign substance. But then, when all immunogens is antigen. Okay, so I hope you can understand the terms. Alright, so then let's go to antigen versus immunogen versus hapten. So this is the slide where I will explain about the three types of antigen determinants. Okay, so basically animal is immunized with these hapten carrier conjugates to produce antibodies. So basically it's an animals, okay, it's like vaccines in animals, so that when we inject with hapten carrier conjugate, so the animal itself can produce an antibody specifics for the three types of antigen determinant. The first one is the hapten determinant. Right, so this is the carrier and this is the haptens. Okay, so we immunize the rabbits with this hapten carrier conjugate, and the three types of antibody that can be produced is the first one is basically based on the haptens. Okay, so antibodies can recognize the haptens, and the second one is unaltered epitopes on the carrier proteins. Okay, so the carrier protein also have their own epitopes. Okay, so the antibody can also recognize the carrier, and the third one is new epitopes formed by region of both hapten and carrier conjugates. So the last one is the antibody that can recognize both hapten and carrier. Okay, so that when we inject this hapten carrier conjugate, the rabbit naturally can produce these three types of antibodies based on the antigenic determinants. Okay. Right, so next, what is epitopes of antigenic determinants? Okay, the first one is epitopes, the immunological active regions of an antigen that combines with the products of a specific immune response. So basically, an antigens, all right? So an antigens cannot bind directly to the products of specific immune response, which is immunoglobulin, antibodies. So antibodies cannot bind directly to the antigens. So it binds only, antibodies only binds to the epitopes that presents on the surface of the antigens. Okay, so here you can see that antigens can have a few types of epitopes, right? So in this case, you have the epitopes like the structure number ones, and the triangular structure epitope number 2, and the circle epitopes number 3. Right? So all of these three epitopes appear in one antigens and can be recognized by three types of antibodies. Okay, antigens are structurally complex with containing many different epitopes. Yeah? So they have different epitopes that appear in one surface of antigen. The immune system usually responds by producing antibodies to several of the epitopes on the antigen. So that's why I just explained it to you just now. So 3 types of antibodies can recognize 3 types of different epitopes on the surface of antigen. Right, next, let's go to the terms of antigenicity and immunogenicity. What is antigenicity? Right, so antigenicity is the ability to combine specifically with the products of humoral or cell-mediated immune response. Example, secreted antibody or the surface receptor of the T cell. So surface T cell ni is cell-mediated lah, and the antibodies is humoral immune response. So basically, antigenicity ni is the ability to ensure that these products, immune response product, can bind to the antigen, basically driven by the epitopes. So if both can combine, the epitopes and the products of immune response, we can call it antigenicity. Right? So there is a low antigenicity, there is a high antigenicity. So if there is high antigenicity, means that the binding between epitopes or between epitopes and the immune response products is higher. Okay, so if a lower antigenicity means that the antibody, for example, cannot easily bind to the epitope or the antigen itself. And the second one is immunogenicity. Immunogenicity is the ability to induce a humoral or cell-mediated immune response. Okay, so if the foreign substance have the capability to induce or to activate the immune response, we can call it high immunogenicity. Whereas there is some molecule, for example, haptens. All right, so if only a single haptens, it can bind to the antibody, but then it cannot induce any immune response. Okay, so all molecules that have properties of immunogenicity also have the property of the antigenicity. Okay, so all foreign substance that have ability to induce immune response, we can call it high immunogenicity. Okay. And also have of course characteristics of antigenicity, because first it must be able to be recognized by the antibody before the immune response is being triggered, right? And molecules that have a property of antigenicity doesn't mean that they have the property of immunogenicity. So it means that, for example, hapten tadilah, even though the hapten can bind to the antibodies, but then it cannot trigger any immune response. So basically, contoh dia, when we talk about handsomes, so handsome basically will be how to say, when we talk about handsome, automatically we know we talk about a male, okay, men. Okay, so we cannot use handsomes for a girl or female. Okay, when we talk handsomes, so basically we talk about men, but then not all men are handsome. Betul, faham So bila cakap pasal handsome, mesti kita cakap pasal lelaki. Tapi tak semua lelaki adalah handsome. Ada lelaki yang handsome macam saya, dan ada juga lelaki yang tak handsome macam kamu. Macam tu contoh kan. Right. So I hope you understand the terms of antigenicity and immunogenicity. Okay, next is chemical nature of immunogens. So immunogens, if you remember just now, is the ability to induce or to activate the immune response. So basically, what is the chemical nature of immunogens? So there is 4 chemical natures of immunogen. It can be proteins, it can be polysaccharides, it can be nucleic acid, and can be lipids. Okay, so protein is basically a very good immunogen. It's very vast majority of the foreign substance is come from proteins. So it can maybe pure proteins or glycoproteins or lipoproteins. Anything regards of proteins is a good immunogens, right? Whereas a polysaccharides, a pure polysaccharides or lipoproteins are also a good immunogens. So because it's poly lah, poly ni like a very long chain of polysaccharides, so it can be a good immunogens as well, like a protein. Whereas a nucleic acids and lipid are usually poor immunogenic, right? So basically, both nucleic acid and lipids doesn't have any capability. Not doesn't have any capability per se. It can induce, but in a very low rate. Okay, so yeah, so this one is nucleic acid and lipids. Okay. So let's talk about factor influencing immunogenicity. There is two factors that contributes towards influencing the immunogenicity, and it can be divided into two. Which is the first one is contributions of the immunogens, and the last one is and the second one is contributions of biological system. So first, let's go to the contributions of the immunogen. Okay, the first contributions of the immunogen is foreignness. So foreignness means that orang luar, something like that lah. The immune system normally discriminates between self and non-self, such as the only foreign molecules are immunogenic. So this one is the one that I always emphasize in the immune response system, where a part of self, if you consider as non-self, you are a foreign molecule, and you are subjected to immune response systems. Okay, and the greater the polygenetic distance between species, the greater structural difference between their constituent molecules. So it means that even though you are in the same species as your, even though you come in the same species, but different phylogenetic distance. So come from the same mother, then you evolve. Then even though you come from the same species, but your phylogenetic tree is very far. Okay, so the greater the structural difference between their constituent molecule, the better the immunogenicity. So means that for example is collagens. All right, collagen display very little immunogenicity, but some self components, example corneal tissue. Corneal tissue basically is a part of collagens. Okay, in the same types of collagens. I mean that corneal tissue is one type of collagen, but generally collagen display very little immunogenicity, but somehow in one parts of a collagen, for example corneal tissue, display a very high immunogenicity. So that it meant by greater phylogenetic distance between species. Okay, right. The second one is molecular size. Right, there is no absolute size range that we can consider, this molecular size is very immunogenicity, this molecular size not immunogenicity, doesn't give any immunogen effect. So there is no such thing. But most of active immunogens tend to have a molecular mass of 100,000 Dalton. So in other words, say like 100 kilodaltons. Okay, so most of the foreign substance size 100 kilodalton and above. Okay, so we can consider it as active immunogens. Okay, generally substance with a molecular mass less than 5,000 to 10,000 Dalton are poor immunogens, with some exception lah. So ada je yang even though the molecular mass is very low or very small, it gives a very high immunogens activity. Ada je, but then with some exception lah. Okay. Right, the third one is chemical compositions. Chemical compositions. In general, the more chemical complex the substance is, the more immunogenic it will be. Right? For example, heteropolymers are usually more immunogenic than homopolymers. So heteropolymers is combinations of a different substance. Okay, you have substance A, substance B, substance C, substance D in one polymer. Okay, and it's very immunogenic. Okay, and another homopolymer is for example consists of substance A, only A. Right, so it tends to be less immunogenic. Okay, so when we talk about heteropolymer, of course, it will involve in secondary, tertiary, or quaternary structure of the molecule itself. Okay, but do remember that heteropolymer is more immunogenic than homopolymer. Right, and the fourth one is susceptibility to antigen processing and presentations. Okay. In these types of contributions, a larger insolubles or aggregated molecules are more immunogenic as compared to small and soluble, because larger molecules are more readily to undergo phagocytosis and process. Okay, so larger lah. Kalau small and soluble, no, it's very difficult to recognize the soluble metal. Yeah? For example, you put a sugar in your coffee. Not coffee lah, coffee is because it's dark, you cannot see the sugar. You put sugar in the hot water, right? Normal plain hot water. So if you just put it without stir, you can see the insoluble sugar. So it's easy for you to recognize. But then when you stir the mixture, so the sugar is soluble. So now you cannot see the molecule of the sugar. Floating around. So it's very difficult to recognize a sugar, a soluble sugar. Okay? So the same analog for the phagocytosis. Right, macromolecules that cannot be degraded and presented with MHC molecule are all immunogens. Right, let's go to the contributions of the biological system. The first one is regards on the genetic factors. Okay, so some substances are immunogenic in one species but not in others lah. For example, you can see here Streptococcus thermophilus versus Streptococcus pneumoniae. So even though it's in species, it's in genus, but then they're different species. So Thermophilus, Streptococcus thermophilus basically is a good bacteria where they use vastly in the productions of yogurts, so we can eat it like that. Whereas Streptococcus pneumoniae is a very virulent bacteria that can cause pneumonia on your lungs. Okay, so very bad bacteria, I guess. The specific individual may lack of have altered gene that code for the receptor for the antigen B cell or the T cell, or they may not have the appropriate gene needed for the APC to present antigen to the helper T cell. So this means that if bacteria A1 can be recognized by the B cell, but then bacteria A2 cannot be recognized. Why? Because they come in the same group. Maybe bacteria B2 already undergo some mutations or altered genes, so it changed the genes that code for the virulence. So so that it can be recognized by the receptor on the B cell and T cells. Okay. And the second one is age. Usually the very young and the very old have the weakened ability to mount an immune response in response to the immunogens. Okay, so orang yang paling muda, like a baby, a newborn baby, or the very old one, like 60, 70 years old, so have the susceptible to the immunogenic foreign substance. Okay, the last one is immunogen dosage. So in other words, like vaccine dosage lah. Alright. Basically, vaccines, one type of vaccine, they use immunogens lah, to induce your antibody immune response. Okay. Insufficient dose will not stimulate an immune response. Okay. For example, when you take the vaccines and the vaccines contain, for example, a lower dose of immunogen, so basically it cannot trigger the immune response. Okay, because it fails to activate enough lymphocytes, or because in some cases certain range of low doses can induce a state of immunological unresponsiveness, okay, or tolerance. Whereas if you get a highly dose of immunogen, it can also induce a tolerance, right? Tolerance. A single dose of most experimental immunogens will not induce a strong response. Rather, booster is usually required. So, kalau you ada adik yang kecil, or you have like, sesiapa yang kecil yang you kenal, and you tengok dekat buku vaccination dia kan, mostly baby lah have vaccination books, and there is a certain vaccine that need a booster, right? So booster 1, booster 2, booster 3, something like that. So it means that they vaccinate you with a small amounts of immunogens, okay? And the consequence, the next vaccine, they will vax you with the booster to enhance the immunogens, all right? In result, it will activate the immune response system. Oh, ada satu lagi. Alright, the last one is root. This is the, the second last, roots. Number 4 is roots. Okay. So basically roots ni, we were going to talk about cara macam mana you ambil immunogens or vaccine tu lah. Okay. So experimental immunogens are generally administered parenterally. So means that routes other than oral. So kalau oral ni, kalau you makan, other than makan, cocok kat mana-mana ke, kat dalam veins ke, kat dalam skin ke, we call it parenterally. Okay. The administration's route strongly influence which immune organs and cell population will be involved in the response. Okay. So contohnya, you nak ambil antibiotics, alright, doktor kata, okay, you have to take antibiotics, but you have to take in the forms of drips, okay, IV. So the doctor will basically introduce antibiotics or antibody directly into your veins, alright, and so it will affect specific immune organs, okay, and if the doctor injects you through veins, right, we call it intravenous routes, right, which is carry first to the spleen. Okay, kalau you cocok anything, alright, tak kisahlah you cucuk ubat ke, you cucuk vaksin ke, you cucuk antibiotics ke, you cucuk whatsoever, even drugs, okay, you cucuk through the veins, it will directly go to the spleen. Okay, whereas if the antigen administered via subcutaneous routes move first to a local lymph nodes. So subcutaneous route means that you inject, but then you tak inject directly through the veins. You inject beneath the skins. Okay, bawah kulit tu you inject. For example, insulin. Okay, so insulin you inject dekat bawah kulit. You tak inject masuk kat dalam veins. You inject masuk dalam kulit. Sebab tu patients with suffer with insulins, dia boleh inject diri sendiri, tak ada masalah. Sebab apa? Sebab dia tak perlu cari veins, dia just inject saja bawah kulit tu. Okay? And the subcutaneous route of administration is desired over the intravenous route due to patient comforts and compliance. So subcutaneous ni mainly lagi patient suka lah daripada intravenous. And the last one is adjuvants. What is adjuvants? Alright, adjuvants are substances that, when mixed with an antigens and injected with it, enhance the immunogenicity of the antigen. Yang ni untuk vaksin untuk manusia lah. Okay, combinations of antigens and adjuvants. And generally, adjuvants exerts one or more of the following effects. The first one is antigen persistence is prolonged. So when the antigen, vaccine ni basically kalau di-inject with antigens or immunogens, either is a weak immunogens or a die-immunogens. Okay, so both weak and die-immunogen ni basically dia non-functioning lah. Okay, the purpose why dia inject yang weaken immunogen ni sebab apa? Sebab dia tak nak bagi a very large effects. Dia just nak antibody recognize je, or B cell, T cell recognize je, so that it can produce a memory B cell and a specific antibody towards the immunogen. Okay, sometimes the antigen, when coupled with adjuvant, it can prolong. So when they prolong, it gives more time to the B cell or memory B cell to develop. Okay, and it's also co-stimulatory signals are enhanced. Okay, so co-stimulations means that, for example, in T cells, alright, rather than T cell recognize MHC class 2, it also have co-stimulatory, so it also recognize CD28. Right, so it means that it will enhance the capability of the T cell to recognize the foreign substance. Okay, and local inflammation is increased, and the non-specific proliferations of lymphocytes is stimulated. So this is the effects of the adjuvants. Right. So next, let's go to the major histocompatibility complex, or known as MHC. Right. So what is MHC? MHC is a set of cell surface protein essential for the adaptive immune system to recognize a foreign molecule. So in other words, MHC helps immune self to discriminate self and non-self. Okay. So basically ni MHC ni is like a guidance of a cell. I know I can say that because it gives a signals to the immune system, basically B cell or C cell. Oh, I'm a self. I'm a part of you. I'm a part of the body. Okay, so please don't judge me, or please don't kill me, something like that. Okay, a major role of the MHC is to bind small peptides and to present them to the cell surface, where the antigen can be recognized by the T cell receptor. Okay, so MHC ni function dia akan persembahkan a small peptide, alright, tak kisahlah immunogen ke non immunogen ke, on their cell surface, so that T receptor can recognize it. Okay, so when the T receptor bind to the small peptide that presented by MHC, so T cell ni dia buat dia punya ni lah, oh, this is the foreign substance, immunogenic, so we have to kill them. Oh, this is not the immunogen, so we don't have to do anything about it. Okay, something like that. So MHC molecule display both self-peptide derived from their own proteins and foreign peptide derived from the invading pathogen. Okay, so last time ada one of your friend asked me about MHC. Does all cell have to have a MHC a complex? Okay, so the answer is yes. So every nucleated cell have MHC. So that is a signals or identifications like an IC saying that I come from your body, so please don't kill me. Okay, so I will explain it to you later in MHC1 and MHC2 types. Okay. Okay. Okay, antigen presenting cells, APC, or known as cell that display antigen complex with MHC on their surface. Okay, so there is two types. There is MHC class 1 and MHC class 2. Okay. Okay, basically MHC class 1s will presented their peptide, short peptides of degraded proteins that comes inside the cell. So in other words, we call it endogenous. Whereas in HEMHT molecule, it presented the peptides, the degraded peptides, come from the outside. So that's how we call it exogenous. Okay, so contoh dia macam ni, ada tak contoh? Okay, right. So this is phase of cell-mediated immunity, antigen processing and presenting of exogenous antigens. So exogenous means that from outside lah. From outside, it's a MH2 class MH2. Okay, so basically MH2 express or present on APC or macrophage or B cell or dendritic cells. Okay, and it only can be recognized by T helper cell and CD4. Ingat so MH2 class ni, MH2 class only presence on the surface of APC, macrophage, B cell and dendritic cells. Okay, and it only can be recognized by T helper cell and CD4. Right, CD4 and T helper cell. So CD4 function dia just untuk check lah. Okay, betul ni MHC2. Okay, so you are granted to further your reaction, something like that. So kalau CD4 react, oh ni kan MHC2, so kita tak boleh nak granted any further actions, something like that. Okay, so for example, why we call it exogenous, because of course macrophage engulf a bacteria from the outsider. Okay, for example, the engulf bacteria and the protein antigens will be captured in lysosomes or endosomes, where the protein is being degraded, and it will couple with the MH2. Some part of the degraded proteins is coupled with the MH2 and will be presented in this on the surface of the, for example, a macrophage. Then it can be recognized as by the T helper cells. So basically T helper cells ni helps to activate B cell to secrete antibody. So bila dah activate, so before they recognize antigens on the MHC2, we call it naive T helper cells. Okay, so after it recognize the antigens or the foreign substance on the MHC2, it become a T helper cells, right? A matured T helper cell. So the matured T helper cell will be function to activate B cell to the plasma cell to secrete antibody. Okay, and it's also help to activate a cytotoxin T cell, or known as killer cell. Okay, so that's the functions of helper T cell. Okay, so let's go to the endogenous. Alright, endogenous where involve MHC1. Okay, so since it's endogenous, basically okay, before that, MHC1s only appear on the nucleated cell, all the nucleated cells. Okay, except red blood cell lah, sebab dia tak ada nucleated. Okay, so basically these normal cells is tend to be a host for infections of the viruses, for example. Okay, so viruses infected. Okay, also it can by phagosome as well. So virus infected, so virus tu tadi secrete up dia punya RNA or the proteins. Okay, then the host will help to synthesize the protein. Okay, so when they synthesize the viral protein, and the cell recognize it as a foreign protein. Okay, so it will undergo degradations by a proteasome. So proteasome ni dia macam pisau lah, degradation machines. So any foreign particle yang masuk ke dalam cell, it will be subjected to the degradations by the proteasomes. Okay, so bila protein tu tadi dah degraded into small peptide, it will transported to ER, endoplasmic reticulum, through a TAPS transporter. Right? Sini ada TAP channel transporter lah. So peptide tadi masuk dekat dalam endoplasmic reticulum. Alright, dalam endoplasmic reticulum dia dah synthesize MHC class 1. Okay, so they bind together here, and when it binds, it will be transported to Golgi apparatus by the vesicles. Alright, vesicles. So from the Golgi apparatus, it will be transported up to the surface of the cell by exotoxic subvesicles. Okay, exo ni transport up lah. Okay, so when it presented on the surface of the cells, it will be recognized by the cytotoxic T lymphocytes or natural killer T cells, right? Along with CD8. So CD8 tadi macam CD4 lah. So dia macam gut yang recognize, okay, this is MHC1, so please proceed with the further reactions. Okay, something like that. So bila it recognized by the killer T cell, cytotoxic T cell, okay, then the T cell will signals this affected cell to undergo a killer cell program, which is apoptosis lah. Okay. So bila dia dah recognize, oh ini viral peptide ni, okay, apa kata you pergi bunuh diri you sendiri, macam tu. Dia nasihatkan sel ni, okay, apa kata you bunuhkan diri you sendiri untuk kesejahteraan badan tuan kita, something like that. So dia pun akan undergo apoptosis, dan bila dia undergo apoptosis, all the virus that been infect the bacteria also will die together, macam tu. Okay, right, so this a summary of the MHC1 and MH2. So criteria on your left side, the first one, irresponsive T cell, is cytotoxic T cell lah, right, killer T cell, whereas in MHC2 is T helper cells. Okay, so types of APC or target cell, MHC1, all nucleated cells. Alright, all nucleated cells. Oh, I lupa tadi. Okay. Kalau means that in normal cells, so they means that normal cell they tak infected by the virus. Okay, what they did to avoid them to be undergo apoptosis. Okay, so their own protein will be degraded, and their own protein will be presented on the surface by the MHC1. Right, MHC1. So when the T cell recognize this peptide, this own the host peptide, so T cell kata, oh ni protein daripada badan you je, daripada cell you. Yes. So we going to do nothing lah. So you are safe, something like that. Okay. So all nucleated cells, and then in MH2, B cell, macrophage, and dendritic cells. and surface of antigens is endogenous, alright, synthesized in the cell, whereas in MHC2, exogenous, internalized from extracellular environments. Okay, sites of peptide loading of MHC in MHC1 is rough endoplasmic reticulums, and MH2 is lysosome yang transport out. Okay, so this is test yourself. You can answer these questions. Alright, so if you're able to answer these questions, means that you are familiar with this chapter 4. Right? Okay, guys, I think that's it for chapter 4. At meantime, we'll put instructions on your assignment 2, which is a group presentations on the GC. So nanti you tengoklah dekat GC tu apa kriteria yang perlu you buat untuk buat second assignment, which is group presentations. Alright? Okay, till then, we meet again. Thank you so much, class, for your time. Assalamualaikum warahmatullahi wabarakatuh. Bye-bye.